Welcome everybody. In my talk, I will speak a bit about how goals can emerge and change. So
yeah.
So first, I will introduce a bit what the issue is, explore a bit what's the problem and the issue at hand. Then I will explain a bit how I think we can resolve this by focusing on changing from within. And then I will present like a little model to explain this even further. So the topic of my of the project and also this workshop is the origins of goal directedness. So first, goal directedness is often like traditional cybernetics account of defining goal directedness is if you have like a lot of participations and you're always going to the same direction. So whatever the input you are able to always go to the same direction. So it's like what's called kind of equifinality. It's focusing on something that is remaining. Well, now we also want to look to like the origins of goal directedness, and here we see how the question is how can it emerge. So it's kind of a bit contradictory because now it's emerging and changing. So often like in cybernetics, like the first view is like a machine where the goal is just given from the outside, put in it by creator, and the goal remains static, so it's always the same. While if we want to consider living systems, here we see that the goal somehow emerges from the system itself inside, in interaction with the environment. And it's a living system is also highly adaptable, so the goals constantly change and so on. So these are kind of if we consider a goal as just something remaining, how can we then still have this kind of change in dynamics? This question. And also, if we look to in humans, if we look to the use of goals in everyday life, we see that we constantly create new goals. Often, like we there can be goals which we like consider like just for one time we consider the goal, and it's like for forever. And sometimes even if something is like a self-maintaining cycle which can remain, it can often feel like a vicious cycle. It can it's like often we don't really differentiate, or it feels the same whether something is if we don't feel like we can influence a certain phenomenon, it doesn't really, it feels the same whether it's external or internal. So also if it's like a discussion I had about free will, they were talking about like internal processes of our body and which we don't really have control over. So these are like internal processes, but still we feel like they've grown somehow, but we cannot adapt anymore. They are not adapted to the new circumstances, to the new situation, or like new goals. So one example is like a certain bad habit you can like keep on doing, or like the vicious cycle kind of things of addiction. Or for example, an example is like sugar. How we deal with like sugar, like when we were hunter-gatherers, sugar was not a scarcity start. So so it was really like if we found sugar, it was really like something pretty something pretty attractive. So our body has adapted so as to really like sugar, so to really like certain mechanisms, so that if you're sugar, we're really liking it. But now it's really abundant, and our sugar still, our body still has this response, but it's no longer adapted to the circumstance. So we are consuming a lot of sugar while it's no longer healthy for our body, but we still have these kind of mechanisms, these loops which create we want to have sugar. And similar mechanisms also happen socially, where we have like social systems which are like can form like a vicious cycle where the social system keeps on existing while while no human inside actually wants a social system anymore. Can be like institutions or organizations, can also, for example, environmental problems. We can see like, okay, we keep on having these environmental problems while everybody may be convinced that we should change and produce things differently. Um, but still somehow we keep on doing the same thing. So now I want to like explain a bit how, on the one hand, these vicious cycles can be escaped, and also how we can have change in a goal-directed way, or like I think that's if the change is coming from within the system. Um, so the idea is to look to like what's considered constraints. So first, I want to like use this term in where it's also used in movement science, where the constraintless approach is like an approach used in like natural movement kind of things. And the idea is the body is capable of self-organizing um, in a and like adapting to certain circumstances, but it adapts to certain constraints. It's so if and you can change these circumstances so that it adapts in the way you want, or like adapts to a new regime. So for example, if you are in an environment with lots of couches, computers, everything made for you to just hang in the couch and do nothing, then your body will adapt in a way that your muscles will deteriorate. So if you don't want this, what you can do is, for example, you just like install a pull-up bar, you install things to move, and then automatically you will move more, and your body will adapt to these kind of things. It will like self-organize to these new constraints. So that's one example of how to use this. So in general, what we can see as a constraints, you immediately think about things that restrict certain things, but the idea is that it can restrict things, but at the same time, this can make other things like possible. Uh, so in general, you could see it as what affects the process. So it makes some things more likely, other things less likely, but which isn't affected itself by the process. So it kind of alters the possibilities of what is possible. So you can see it as like a catalyst in chemistry, or which is like happening in the reaction, but it's like not being consumed by the reaction, or like a blood vein, or it's also an example of it, which can also be like it's like a pipeline structure, and it's like on the one hand it's really constraining the things, because like things cannot move random anymore, they need to move in certain direction. In the other way hence they enable certain things can move to places where they cannot go anymore. You can see um, it's like a pipe connects certain things. So is in general constraint for this to be like autonomous. The idea is that this constraint comes from within, so it's produced by the system itself. So that's why the idea of a closure of constraints comes in, where the idea is that all constraints are produced from the system itself, and each constraint is also producing or like affecting another constraint in the system. So you have this kind of cycle loop, kind of self-maintaining structure of constraints. So, for example, if you look to a blood vein, on the one hand, it enables other things in the body. On the other hand, it needs this body to be produced, to be able to exist. So I think we can use this kind of this idea to see how change can happen. So if you look to the super example, what we can do is like we can decide based on some trigger, some information from the environment. We as a complex organism decides to like not buy any sugary food anymore. So for example, we like decide to go on a diet, not buy any sugary food. So that's like a new constraint arising. And then what can happen is that the body adapts, so that this sugar craving cycle is like diminished, and there are like new hormones produced, so that we're like we don't consider something really more sweet than before, or taste change and so on. So there's like a production of a new constraint, and because we are like not that interested in sugar anymore, we will also like have less the urge to buy sugary food. So we are reproducing the first constraint. I think this looping back is also important, because if you only have the first part, what often happens is like first you're motivated to like not to have this constraint. You're motivated to like not buy any sugar food, but like if you're still sugar craving, you will on one moment you will start buying it again, and this won't be able to sustain to be self-maintained. So like social change is another example where social change can happen because people do certain actions which result in certain changes, and these changes result in people wanting, or more people doing actions, and so creating change. I think often the thing with social change is that it cannot loop back. So people are first motivated, they want to have change, they do certain things, but then they don't really feel it results in certain things. It doesn't produce actions again. It doesn't loop back anymore. Then you get things like activist burnout, people who like start being motivated, but then afterwards they become less and less active. So to conclude, it's what I'm saying is that instead of like focusing for world happiness as like something that remains despite perturbations, I think we should try to focus on that it comes from the system itself. So in a way, we could differentiate goal directedness as focusing on like what remains, and then autonomy as also that it comes from the system itself. Because like a machine can also be goal directed, but the goal is imposed from the outside, so it's not autonomous. It's not coming from the system itself. So I think it's better to focus on coming from the system, because it's focusing on what that it should remain certain things. It's like neither necessary nor sufficient, because like the classical example is like a ball falling into a valley. It's pretty stable, but we wouldn't call it goal directedness. Then often people say there's another requirement necessary, like it should be far from equilibrium. The system should act so as to go to this goal. So I think this extra condition is actually saying that coming, what you can say, from the system is acting to go that direction. And then it's also not necessary, because goals can also change. There can be like living systems are constantly adapting, and you cannot really say that something is remaining, but still we would consider it as like goal directed or autonomous. So in general, what happens is that the system acts as a constraint on itself, so it's altering the possibilities of what is possible. So one possibility is that the environment moves everything in random directions, and then the system can move in the same direction. So that's like the classical idea of like equifinality. There are different inputs, different perturbations, but the system is able to still go into a certain direction. But then it could also be that actually the environment is always it's like moving to the same direction, but depending on the system, depending on internal circumstances, depending on external circumstances, the system is still able to move into another direction. For example, we can consider gravity, where a non-goal directed system, you would have like a stone which is like always falling to the ground. You have like just an external force which moves the things, while but if you have like an organism, a living system like a plant, it can grow up. An organism can like move into any direction it wants. It like uses these forces, so they're like not putting these forces out, but it's using these forces to somehow constrain these forces so that things are tweaked like in the direction it wants to go. And so why is it possible, this choosing your own path, this self-causation? It's because of circularity, where things can go back to each other, so then you can cause yourself and you're like less determined by the external. And then this change of path is it can be constrained by the system, where there's like this new closure of constraints, this new self-maintaining emerging. So also this view makes that we cannot really distinguish one core, or this like we cannot say there's like one goal or core goal. There's more like a coordination or different processes which somehow align, different interactions which align, and there's like a direction coming from this alignment. And at the same time, so these interactions say these, you can say it as different goals which produce each other, but at the same time can constantly change and kind of really. So this is like the general idea of the theory I have. Now I'm going to present a little model of how we can see it. So first you can like, we already consider we have a certain system, like for example, like a chemical organization. Like this is kind of a rude example of like a simple chemical organization which has like a lot of things inside. So then we would say there's like an external cause if there would be like a process from outside which like transforms certain things into other things, can like maybe use consume certain things from the organization, can produce certain things inside the organization. So this organization is altered from purely from the outside. And then internally caused would mean that there's like this process only happens when it's like constrained from the organization. So there's like catalysts from the organization itself, which is like enabling this reaction to happen. So this reaction only happens when this catalyzed, when this organization is there. So it can be like some things inside organizations which makes these things to happen. So it might seem like a small difference, but I think still it's like a big difference, whether, because now it depends on the organization whether this process happens or not. So I think it's a bigger difference than you think. And so then this is like the first part where it's like internally caused reaction, but then what we saw is this kind of still like just disappear again. So we had like, for example, the sugar thing, we had like, it could be that we had like a trigger which motivated us to not buy any sugary food anymore, to buy more healthy, but then after a while this trigger isn't there anymore, and we start up in the same cycle again. But so this kind of humane if there's like closure of constraint produced. So for example here, that's like a most basic example is like this Y, it's constraining the production of a certain Z, and this Z is constraining the production of Y, so that that's as soon as Y, as that what we have the production of Y, this thing can be made. And so this you can see this closure of constraints in this kind of way. Maybe it's just like self-maintaining new self-maintaining regime can also be just like another example. So in what way exactly this is open. So for example, to explain this concept a bit more, take the change in the super cycle. So what we at first had, we had a certain trigger by catalyzed constraints by the organization. So because of our internal structure or goals or values and so on, we produce the constraints of not buying any sugary things, and then this constraints produced us producing the constraint of like a disinterest in sugar, certain difference in hormonal or kind of bodily stuff, which produced the a disinterest in sugar, and then this constraint again produced the production of constraint of not buying food anymore. You see here how it can like get incorporated. So these are pretty like human high level examples, but I think the exact same mechanism applies also at really pretty simple biological levels, like really origin kind of life kind of things. So here I give the example. So I want to the idea is just to illustrate how this can apply here. I'm not a specialist in biology, so it might be that there's certain details which aren't right, but it's more like to illustrate how this can work. So the idea is to so the idea of a plasmid incorporation is you have like a certain bacterium and their plasmids, so they're like certain DNA, RNA kind of things which are floating in the environment. And the bacterium decides to take in certain plasmids, maybe not take in other plasmids. And so constrained by the bacterium, certain plasmids are incorporated inside the system, so there becomes like an integrated plasmid. And now this plasmid can like fulfill certain functions, so it can trigger certain processes to happen. And then because of these processes, maybe the bacterium will survive more. It might, in general, this newly acquired plasmid will be reproduced. And in this way, this can be incorporated and like internalized inside the system. I think the RNA world can also similarly be expressed in these kind of things in a way that like the RNA can also be in certain ways seen as a ribosome or transformed in a ribosome. And then this one can somehow produce nucleotides. And then the RNA can actually like reproduce itself. So because nucleotides can make like a inverse RNA, so like the inverse strand of the RNA, and then this inverse RNA can then again reproduce the same RNA. So this way you can have like this RNA constraining the production, constraining itself to be produced. So it's like coming from inside the system. So this is like so here what we saw was like we saw constraints morally purely as like catalysts so catalysts defined in chemical organization as like a certain species which is like present inside a process, so it's like making it more or less likely to happen, but it's like not consumed or produced into the process, so it's like staying the same. We already, like I already said, like can be really a catalyst in the chemical sense. We can also see it more like chemical organization in general, more broadly as like, for example, a blood vein, which is like affecting a process which is affected by it. But we can also even more general try to broaden this concept of constraints. For example, we could also see like a resilient organization as a kind of constraint, because what it does is like there's a perturbation coming from the outside. It can like neutralize this perturbation. So the organization isn't changed, but at the same time, what happens with the process is different, because now its perturbation is neutralized, while before it was still there. So that's another kind of constraint. And so in general, we could see it as whatever isn't affected by certain processes, but is affecting them. So we could see it as like more like a set of species that is constraining like a set of processes if the dynamics, the concentrations, everything inside of this set isn't changed whether these processes happen or not, and the reaction rates, they do change whether these sets are there or not. So for example, I think we could also in this little picture see the cycle of A going to B and B going to A again as a kind of constraint, where if this cycle isn't if this cycle isn't there, then X won't go to Y because X needs to interact with A to be able to produce this Y. So the process X to Y is only happening when this constraint is there. And on the other hand, actually this AB, whether this X is there or not, whether this process are happening or not, it will still be able to cycle to exist, because like this B is also produced again, and then automatically the system will find a new equilibrium where A and B becomes like the same concentration again. And like I think we can broaden this concept of constraint, but it also makes it more difficult to see like, oh, where are there constraints, where are there constraints, to recognize when is it a constraint, when isn't it a constraint, and then also so also I spoke about like that it's like affecting so it's affecting a system, not just producing. So it's affecting certain processes. It's not only producing it, so it can also be that it makes things less likely. So that's in this way, because often with autocatalytic cycles, the critique is that there can be like positive feedback of production of more and more and more catalysts. While but if you consider not only producing but also like consuming certain catalysts, then what you can see is that they can like that things can that also certain catalysts can be diminished, and so they can become less stable, not go indefinitely. And then also this, so we can also broaden the concept of closure of constraint. Like closure of constraint was defined as each set of constraints where each should like affect another constraint and be affected by it. But the main idea was mostly that the constraint was coming from the system itself, so it was produced from the system itself. It was somehow affecting its own production, its own self-maintenance, which is in one way not always the case with this definition of this previous definition. On the one hand, it can be in certain cases maybe it's like affecting its own production, but it's in a different way. So it's like needed for its own existence, I think. Like the main thing. To conclude, what I talked about is first that rather than looking at like what remains, we can look to like how change can happen from within, and that's when the organization can affect certain processes, so alter the possibilities, and then this change can be internalized by so it should be like self-maintained by having this so this constraint should form a kind of closed kind of thing.
Yeah. Thank you, Evo. We have like a couple of minutes for questions in case there is anything. Pasquale, please. Hi. I think we cannot hear you well.
Okay, can you hear me now?
Yes, now
Okay.
it's good. Yeah.
So yes, thank you for the presentation. I agree with what you have said. And my question is, this constraint closure is a necessity in a dynamical system, or you can imagine a dynamical system without constraint? Because for me it's difficult to imagine. So it's something that comes together with dynamics and with systems where elements interact with each other in different ways. And I don't know, what do you think? Is the emergence of constraints there? Because
Yeah, I
it's
think
intrinsic in the system. Of course, the closure is maybe is a particular case where if the system has also closure, then it goes in a very specific dynamics. What do you think?
yeah, I think like what you said, there are always constraints in the sense that also just certain things are more possible than other things, but like there can be like external from the system, where they're like often in a dynamical system there are like external constraints which makes the system move in a certain way, or it can be like internally where these constraints are produced from the system itself. So to have this case where these constraints are produced internally from the system itself, I think it's a pretty specific case.
Yeah, I think even in simple molecular systems like an enzyme, the constraints are they determine the folding shape enzyme, and therefore, in cascade determine which kind of molecule can bind. But ultimately, this is due to the impenetrability of matter. So when you have a long string, you can fold, but you cannot, cross, occupy the same space, and therefore, in every level there is a constraint. Yeah. Thank you.
Well, thank you, Evo. We now move to the next talk, which is Olaf. Olaf Witkowski, who's visiting and will give us the next talk. He's now taking position. So.
Good.
People can hear. Maybe someone could confirm that it hears well. Yeah. Okay. Great. Thank you, Barbara.
Okay. Might not the camera just.
No, we can arrange this.
Okay. Amazing. All right. Okay, this talk is a little bit different. I guess I'm not talking COT, but I'm touching on several of the topics that Francis, Thomas, Evo, and Peter have been touching on, because I'm talking about intelligence. And I'll zoom onto three things, which is control, essentially, perception, and care and goals at the end. So those are essentially three little projects. Here they are. Quick self-introduction. So as I come from Japan, I'm visiting here, CLEA in Belgium. Very happy to see everyone in real life here. So what we study is actually intelligence science, and we couple that with technology. So there are different aspects, I guess, of intelligence, and we try to focus on everything. So we zoom onto different aspects. These are our kind of flagships real quick, because I don't want to spend too much time, but collective intelligence, I know that Francis is a big fan of those kind of distributed intelligence approaches, open-endedness, and then mathematics of what is intelligence effectively in an embodiment context. So ALife is a big kind of focus, so we're happy to be at the conference. Maybe I'll skip a little bit on this, but essentially we are coupled very closely to the industry to find wherever there are ways to change the way things are done, not waiting for 10 years before an application. All right, so this is who we are. And maybe that's a thank you for the team. These are the directors of the institute in Kyoto, and this is the team. I thank everyone in advance, so I don't forget that after. All right. Maybe quick flashbacks. A lot has been said already about cognition, about different aspects of goals. I really like the relations with evolution here. And yeah, what is goal directedness in different ways, from the very low scale of minimal cognition all the way up to human and different types of intelligences. I really like that. That actually touches a bit on this first project. So I jump into it. I'm giving you the conclusion. So if we don't have time, this is the only slide that counts. So after that, we can play around. So the first point that I want to make is about dependency on substrate, but really the fact that intelligence, as we often mean it, as in the mind, may vary in its instantiation over various universes. And if you change the laws of the universe, if you change the chemistry, right, your network, but not only that, but parameters, maybe the intelligence means something completely different, also in the goals that, that emerge in there. Second is more zooming onto the perception aspects. So if you study if you're interested in the phenomenology, right? So how people report about how they experience different concepts, how they see something like colors, which is what I'm going to focus on. This may vary very widely between cultures and also embodiments. And this, so I'm proposing a sort of approach to study that. And last one is a very fun project. See if I can move this thing so I can see my screen. No, I don't see my mouse, so that's hopeless. All right, I'll guess what is behind it. I can see it there, which is care light cones. So, the difference between the light cone of a sort of a state per se of an agent and its relation to the space of its goals. So what's the mapping between the two? And I'll link that to Buddhism and AI. So if you stick around, you'll hear about it. If I haven't scared you away yet. All right, first project. This is the one that I worked on with Martin Beal. So a little picture to honor him. He would be very unhappy that I presented this way and so quickly, but here you go. So what we're interested in is here a very simple idea. Is more compute meaning more intelligent, more powerful, right? So if I add compute into your machine, does it mean it's more intelligent? And of course, if I say it this way, the answer is no, but let me show you why. So what we are going to use is cellular automata, it's written over there, and we're going to define a sort of volume that is within an environment, right? So it's at the center here of an environment, but any surface of contact would be okay. This is just an example. And there is a space Z that embeds all of that. Now, the environment, I mean it as a controller of a certain machine. So you are using this calculator, for example, you are controlling this machine. So E is controlling V. Right? And what we use as a model is, you've seen this before. If you haven't, then definitely attend more talks at this conference. This is cellular automaton, right? This game of life kind of thing. We are going to not even go into 2D, but I'm showing you the 2D, which requires more computation. Any more dimensions are going to be very interesting as well. What we can do here is define this volume, right? This is the computing stuff. Your calculator is in the middle. Now you have an environment that surrounds it that tries to control the calculator to produce operations that are useful, right? So you're trying to accomplish a certain number of functions. And we have an algorithm here. I'm not asking you to read everything. It would be cruel. But essentially, what we design is a way to count systematically how many functions you can squeeze out of the inside, right? The volume inside. And if there are more, you're happier because you have more potency in your calculating volume. And that's exactly what we plot. Sorry, it's a bit small, but we plot volume against size of environment, right? Size of the volume inside against size of environment. And we count the functions, right? That's the vertical axis. And you see there right away that there's a peak and it shouldn't be there, right? You expect that with more, a bigger size of the volume, you always increase the number of functions that you are able to compute. And it's just not the case, which was very surprising. There were other surprises, but I want to focus on this one, right? So we are counting, again, those functions for different laws. So different rules of the CA. That means different laws of this universe. And again, I know the CA is not a real universe, it's so different, and so on. But you know, this is a tiny step. It's very surprising that we would have, maybe I'll show the next one, on this one, the sort of number of functions over the environment sizes, thicknesses, we call that, and volume sizes for a certain rule seems to peak and then it goes down again. So it's very strange. So your computer, let's say you add more RAM, more compute, more stuff in your computer, you add more, and it's better, and then you add more RAM, and then it starts going down again. Isn't that surprising? Well, that's what you get for weird rules, I guess, of your CA. All right. So this is the kind of small conclusion of this part. So somehow the controllability measured by this number of functions, right? We're not really measuring controllability, which has a very nice literature. I link you to the paper. It's very exciting, especially the nice work by Yansing and Scott Aronson were amazing on this recently. So a small part of this is here. We're just counting functions, and somehow this control may drop with the size of the computing stuff. All right, this is one. Breathe and perception. I love this because I'm really interested in phenomenology in general, and we were interested in how to make those approaches computational. And we focused first on color, and that's often what you hear, right? The redness of red, and what is describing some experience and seeing whether other people feel the same, and whether you can map your experience to another agent's experience. So there is a very big debate about how many dimensions there are in our color perception. And often it's, I don't know, in the room, do you know like what is being said? Yeah, is it, yeah.
Yeah, the typical RGB.
2, 3, yeah. 3 is the usual. I'll show a slide. Maybe I'll show it right away somewhere. I don't know, it's not here. But yeah, basically it's usually 3, right? RGB, your thing. But actually there are other ones that are more physical. I don't mean seeing in 3D here. I mean seeing color in 3D, right? So it means really visualizing color. So this is if it were sound, right? Like phonemes, it would look something like this. And we would count formants, and this is a sort of sphere packing study that we did. But it's sort of similar, right? So what's the optimal way to somehow store is one question. It's a side question. Your colors or your sounds, right, within your mind. So how do you organize that? So you could also think in 3D or an ND, whatever the dimensionality. How do we find out about this, right? So I want to insist. We don't focus on physical or physiological. We focus on really the mind here, right? So just precising that is the usual example. It's a bit, yeah. I'm kind of utilizing this a lot. I love this. It's not completely related, but if you compare A and B, have you seen that before? Is it the same color? So yeah, you probably all know it in the room. It looks like it's different. Actually, it's same colors. Okay, I'll do it a few times. Looks different. Same color. Magic. It's magic. So this shows the difference between that. There's also an important difference here between the sound space and the color space. It's all in your mind, but of course, it's mediated by some substrate, which is very interesting. I want to insist on that here. Skip to this, which is what we mentioned right here. It's often, we talk about hue, saturation, and brightness or lightness. For this, there are other ways to do it, often 3D. Again, that's within the physics. But you do it also, you transpose that into perception. So we tested that. Okay, so this also has a role in culture. Very impressive that always the colors evolve. We talked about emergence, right, of goals today. Or that was the focus. So the emergence of colors seem to happen often, very often in the same order. And I know that we are all humans and we have all very similar substrates, but it's still surprising that they appear. You know, if you have 2 colors, it's black and white. If you have one more color, it's going to be red. One more color, it's going to be, and etc. So there's a very nice paper. I point you to that. I can post the link later in the Slack. That's right. So I'm skipping over this because this is our experiment. What we do here, maybe I'll show it on the other slide. How about this one? It's tiny, but essentially I'm showing you 2 colors, and then I'm giving you a third one. I'm asking you whether it's closer from one or the other. Right, so I give you a sort of bluish thing, I give you a reddish thing, and then I give you an orange, and I ask you orange, this orange. I don't tell you the word, of course. I show you, and then I'm asking you to put the slider. Is it closer to this one or this one? So this helps me compute a sort of a measure of distance, right, between all those points for one mind, right? You can average it. You can do things for a population later, but we have this sort of metric thing. It's not a metric, but let's call it like that, which has very interesting properties. And based on that, after that, we minimize the error between between the different samples that we have to minimize the sort of clashes of distance, right? So imagine that you have this multidimensional graph, right, in n dimensions, and you try now to somehow, you have values for each link, but there are too many links. So there are some kind of spring, so you have to force it into this, and you minimize the energy of that, and that gives you a sort of solution by minimizing that, and you plot how many dimensions it gets you to minimize the energy. Now, of course, more dimensions is typically going to, it's not proven yet, a friend of mine is proving that, but it's going to make you drop. But we actually are looking for elbows, and this is not for perception, this is for sphere packing theory. How many spheres you can pack with of the same diameter within a certain volume. And this is this is arguably very similar. Right, no more on this because I took a bit of time already. So, but essentially, it does point to sort of between 2D and 3D dimensionality, depending on the approach that you take. And not making too precise, but my point here is that we can look like this, with methodologies like this, we can look for invariance, right, in perception spaces across different types of intelligences. You can do it, you can do the same thing with artificial agents, which is sort of my hint here in this conference. All right, I took already 14 minutes. Let's show you the third one, which is very exciting. So this started, so again, collaborators, I didn't name the other ones, was Javi and Onelis for the second study. Here I thank Tomas, Bill, Lisa, and Mike for this collaboration. This is a study essentially of care and state space. So how do those relate? And it starts from us having this project about apparent selves. So we want to understand what the self is, especially between ancient traditions like Buddhism and AI. We had actually a workshop on that last year at ALife here. So some of you might be familiar with this. And what we are trying to see is commonalities, right? So we're trying to map, because we talk about the same thing, sometimes with very similar words or the same words, but we sometimes mean very widely different things, but vice versa. There are sometimes mappings that can help each other for those fields. And essentially that's what we're doing. Sorry, no time to read that. This diagram, right? Biology, we want to relate what is a biological being with whatever the space of AI makes us understand about what an agent is, and what Buddhism tells us, right? And we try to decipher what care is about. And it's very similar to goals for the purpose of this talk. All right. So if we look at this, now we see here in the middle sort of what we call those light cones. Now it's very weirdly shaped cones. The vertical axis is time, and the horizontal space or axis is space. And essentially, we try to plot different creatures in this context and see how they relate, especially if they are in the same space shared by different agents. A bit of water. All right. What is really fun is that if you don't only plot the goals, but also the states here. So states here are going to be in yellow, and unsurprisingly, they will look, I'm plotting a light cone like in physics, right? If you're familiar with whichever theory you are into, really you are stuck into a light cone because you can't move far away fast enough. You can't have an influence fast enough, right? So you are in sort of light cone of influence onto your environment. And in terms of the goals, though, you have an influence over this sort of inverted cone area. So that's what we call CLC, and so that's the care light cone, whereas the PLC is the physical light cone, which is the state. And there are clashes between them. You may be attracted to states that don't exist. Let's say you play something. Do I have that slide? I do. That's for you, Tomas, right? So this is Go. It could be chess, right? Your favorite game. You're playing a position of of Go, and this position may not exist. It may not be reachable, right? It doesn't matter. You may aim at it. So it's part of your blue light cone. It might be completely out of reach, but still you can use it as a goal, right? So your goal may be expandable. You may expand your goal. This is just an example, right? But because I plotted it tiny, it could be much larger and could extend. And that's what we are talking about in the paper. Through something that we call Bodhisattva vow, you can extend somehow your goal light cone. And I'm not explaining the details here, so sorry to be hand wavy. Happy to answer questions. There are different light cones for different types of creatures. So again, the same ones that I showed before, a human, and through the Bodhisattva vow, perhaps something like this. So there are also different, you could focus, and we can make an agent-based kind of modeling of this and see how a statistical distribution, what it would look like, and how it would affect you playing some very simple game like tic-tac-toe or chess, and see what the effect is. All right, so this essentially, this study was about this translation between different theories there, but it's also this mapping between the state and the scarce space. I didn't give the full example of the Go game. Perhaps I should do that and share later. But essentially, this is the last one of the three. So just to recap real quick. So if we focus on intelligence as it could be, rather than intelligence as we know it, we're trying really to find what are the degrees of freedom that we can play with, to investigate how control can change with increasing, say, sizes of computation or different rules, right? CA rules correspond somehow to your rules of your universe. Secondly, your perception space may be completely changed by somehow tweaking, switching to a different culture simply, or maybe going to an animal and asking somehow those same questions, or an artificial agent, right? So we could do that as well. And the carrot light cone is this transposition between just simply your state space. It could be just the state of whatever you are simulating your agent on, and the goal space, which is often represented as equal, and you can, but you have to remember that they relate to each other and have a weird mapping, and including that mathematically, I think, is very valuable. All right, this is about it. Thank you. Happy to, I left some space for questions. Can also go into details if we prefer.
Okay, thank you, Olaf. Thank you for your talk. We see if there is anybody, people clapping, any questions?
Yes, if it's
Peter.
possible to ask.
Yes, sure.
I was quite fascinated by the first part where you showed this functionality analysis in the cellular automaton. So it was in the game of life and other 2D automata, right? So I wonder, I mean, I have basically two questions. So have you also done the same in elementary cellular automata? Because they're, let's say, it's easy then to compare with others. And is it somehow related? Maybe the second, just put the second question. So do you know this work by our host on the computational hierarchy of elementary cellular automata by Barbara Hudokova and Tomas Mikolov? I just stumbled across this. I also found very interesting. Is there a relation between these two works? And if yes, how are they related? Because it seems a very similar aim, right?
Yeah, very good. So first, I'm busted. So I wanted to simplify too much. I didn't compute it in a two-dimension 2D CA. It was too costly. So we did it on elementary CA. So we did it in 1D.
Okay.
And Bara, indeed. So we cited your work with Tomas. We cited your work in the study. So I think it's different approaches. But yeah, I think they are related, of course. So there is complexity on the one end, and we didn't study complexity at all. This would be a nice extension. What we studied is just counting different functions that are not isomorphic. That's it. Bara, maybe you have a comment.
Yes. So from what I understand, it seems like we just chose different approaches of how to measure the computational capacity of the objects. And I just got an email from Olaf pointing at this paper, and I'm really excited to read it and see if there are any correlations between the results. And yes, so thank you. This was a really cool presentation, and it seems very relevant to us.
Yeah, thanks for the question, Peter.
Yes.
We have time for another question. If anybody here, Francis?
Yeah, you might as well.
Well, I'm particularly interested in this idea of the care light cones. I already looked at that in the work of
Shima. I know, that's not it.
Levin.
Michael Levin.
Yeah.
Oh, yeah.
Michael Levin. But the care light cone is, as far as I understood, the kind of things that you are somehow aware of, that they have either happened in the past or they exist somewhere simultaneously in space, or may exist somewhere in the future. So I see the connection with goals, but is it exactly the same?
Yeah, so I name it goals, it's not, right? So I made it a bit simpler because we are talking about goal directedness here. It's not, except for this example of, for example, playing chess, right? But here we really mean it. In the paper, we really study what you care about, and you can care instantly about something that is very remote in space that you don't know of by just isomorphically looking for it. Just you can think about it, right? So that is what it is meant. So you can care about those remote in space and time and distances in the goal space, right? Which is different from your state space. So you're kind of removing yourself from those constraints. There are perhaps some tricks that you can use there that are not elementary, that we don't consider normally in modeling, that allow you to somehow jump, maybe some kind of category theory jumps into somewhere and back to demonstrate it with less computation. So I mean that's a wild dream, but yeah. So it's different. It's different from.
Well, I actually originally understood it in the work of Michael Levin as a kind of a cognitive horizon, and that's why a tick, of course, has a very limited cognitive horizon, while a human, in principle, can care about the Big Bang and about the end of the universe in 1,000 billion years. Mm-hmm. So then it's more a question of somehow quantifying the extent of consciousness or the extent of cognition. So in that sense, I think that's a very interesting concept. Yeah. And maybe in our work with reaction networks, we may find some equivalent. But a priori, the Maturana and Varela approach is, of course, that you don't really care about what's outside, you only care about what happens to you. But higher order cognition will, of course, create shortcuts so that it anticipates things that are outside, because they will affect sooner or later you, and that's where the goals come from. You can have a goal that's very far away from you, but you can still start working towards
Interesting. It. Yeah, absolutely. So so I can't talk for Mike, but I'm fairly sure that he meant this horizon. So this is why this work is distinct. It's also with the same Mike that we worked, just to be clear. So this is really an extension. So we are not using exactly the previous model. Now, it might be that, as you said, right? So the constraints that you would see from Maturana, we wouldn't have those kind of like limitation of scale. It's possibly there is no constraint that we see so far, right? So in our modeling, this stretches right away into all the way across the universe if you want to. So in your state space, there is no limits. You navigate in this alternative space, which allows for some tricks. But now, we have.
But it's a conceptual space, which means it's a model of reality, and you don't know whether the model is correct. And that's what Maturana would say.
All right.
You don't know what a real reality. You're actually traveling in an internal model, and that internal model may have nothing to do with what really happened 10 billion years ago.
Yeah, so we have a longer chat later coming up. I don't want to insist too much, but so it's true. What you say is so for ALife to ski around, it will be a model of reality. In our theory, this is very distinct. So we go, that has more properties than being just a model. So it's not just coarse graining or anything like that. right? And it's not, doesn't have to live within the state space, right? It's distinct, and maybe it could be enclosing that space, for example, or something else. So we don't take assumptions. We just we distance ourselves. We epoche ourselves away from that, and then we see where it goes. So we don't take that assumption. But it's true. If you take that assumption, then yeah, that's done. Yeah, we are limited. Good question.
Yeah. So we leave definitely more time for discussion later, but for keeping you need to stop sharing the screen.
Yeah.
So we can share the last talk of Daniela Flores. It's coming up now.
Maybe after my talk we can discuss a little bit more.
Yeah. So it's you can take position.
Yeah. All right.
It's a PPTX.
Yes, I think
Yeah.
so.
There is only one slide that doesn't look really good, which is this one, but it's just the beginning.
You
Yeah.
can change that.
Yeah.
And the second one, I think too, or no, but it's not a problem.
No, it's okay. No, it's just the first.
so
Okay. So we will share your screen now.
And how I can use the keyboard to change.
Wait. I'll tell you. Let me just select. Start from first slide, and now zoom, share screen, just do share. Okay, so you control it with this, or you can go page up and down.
Oh, okay. No.
Yeah. Yeah.
Okay, excellent. Okay. I am Daniela Flores. I am going to present this talk, Origin of the Form, Patterns, and Homology, What are Goals in Vertebrate Evolution and Embryology. My background is more biological, so in general, I think that this talk will be more related to biology than to the previous talks. Down here. Sorry. In this talk, principally, I think that I am going to be very fast, but briefly, I am going to speak a little bit in the first part about evolution of life, specifically about vertebrate evolution and some processes and forms and patterns too. In a second part, I am going to speak about homology, some concepts related to homology, deep homology, and organizers during development. And I am going to try in the second part and in the third part to relate some processes during development and that have an evolutionary background too. With some concepts of dynamic adaptive system that we have been talking a little bit about that during these talks. So first of all, related to the evolution of life, one of the most important view of evolution now is the extended evolutionary synthesis. And that specifically relates, not only focusing the organism and focusing the organism more in the relationship between the organism and the environment, but not only by natural selection, but also about other kind of processes like developmental plasticity and developmental bias. And obviously, this is a a more complex process that affects in different way, in different levels, different processes in evolution. Like, for example, here we can look the process that generates novel variation, processes that generate bias selection, etc. And we are going to focus more in this kind of processes, of developmental processes. for example, these kind of processes that generate variation during the development, and then that contributes to the phenotypic evolution. In relation to the vertebrate evolution, we know that our ancestors, we developed from bony fishes, that one of them, the vertebrates in general, arose around the Cambrian explosion. And we developed from lobe-finned fishes that then we know that they generate the main group of tetrapods. First of them were amphibians, and then the other big groups of tetrapods, the amniotes. And in general, the vertebrate, all vertebrates, and they have a basic body plan. We can look a little bit of that in the vertebrate body plan, that, for example, a segmented brain, some fins, branchial arcs. This is very important also. And other structures that developed before in the chordate body plan, but they conserved and they are present also in the vertebrate body plan, like for example, a digestive tube, obviously a heart, an otochord, etc. And specifically in the case of tetrapods, that we refer to tetrapod as this kind of organism like ancestral amphibians that arose from lobe-finned fishes, that this kind of fins, lobe fins, developed in different limbs, that we look that they are different in morphology, but they also have a lot of things in common. We know also that our evolution, we can take some aspects of our evolution through the development. There is a very well known sentences from Haeckel, that he said that the ontogeny recapitulates phylogeny, and he refers to that that development during development we can find some signals about evolution. Obviously, not all the process that not all the morphological change, for example, that we look in evolution, they are represented in the same way during development, but a lot of processes and forms that we look in evolution, we can look that during the organogenesis, for example, they show the same pattern. And but also during the cleavage, for example, and gastrulation, despite in general during evolution we look that there are some forms, some patterns that they are in some lineage evolution, which we look that there are some patterns that there is a tendency to develop some patterns. Also, we look that there are some phases during development, like cleavage or gastrulation, that they are very variable. Like, for example, this is something I want just to show this very briefly. For example, in the case of mammals or amphibians or birds or reptiles, the first phases, the cleavage phases, or this first clonal reproduction of the first cells, they are very different in their morphology, in their patterns of production, and also in related to the movements that the cells have inside, not only during cleavage but also during the gastrulation, that is the second big phase, big stage after the fertilization. First is cleavage and then gastrulation. During the gastrulation, there are a lot of differences in the movements, in the cellular movements. For example, radial intercalation, directed migration, involution, invagination. But despite that, there are, during the gastrulation, and despite all embryos, for example, in the case of ancestral chordates or ancestral tetrapod or an hypothetical amniota, etc., reptiles, birds, we look that all the embryos in general, they show the same fate maps. So despite these early differences of processes, and in the case of cellular movements also, which we look that they converge to a specific state, like stages like gastrulation, that they are in general pretty similar. And despite the cellular movements, they are different. They generate something that we are going to discuss a little bit more after, organizers that they have all in common. For example, one thing that I wanted to show here is that they are very different. They are very similar in the fate maps, but one thing that change a lot between the different lineages is the amount of yolk, but it's something that, but the fate maps are conserved. So, and after this gastrulation, these early phases, we look that all embryos, vertebrate embryos, in general, they converge in some stages that they are not in the different vertebrates. They are not specifically the same, but they are very similar. They develop the typical structure of this phase that Hegel, for example, defined it as a philotypic stage or a pharyngula stage. So we look that during embryology, we have a directionality. We can find a directionality of the development that is related to developmental time, but also an equifinality of the forms, not only during development, but also during evolution. And I am not going to speak a lot of this, but there are a lot of, obviously, processes that, for example, acts before the philotypic period or philotypic stage, and then a lot of other processes that affect the form after the philotypic stage, and that produce the variability that we know in the different vertebrates. For example, developmental constraint, peculiar modularity of developmental signals, a lot of things. And also something important are the morphogens, enhancers, for example, gene regulatory and gene expression network, etc. Morphogens, for example, they are molecules that we are discuss a little bit more about this, but they are molecules that, depending on the gradients that substructure is exposed, the structure is going to take one identity or another. Enhancers, they are like promote some expression of gene of some specific genes and so on. So to continue, I think that it's important to focus in some concepts like what is homology. And I started to speak a little bit about deep homology. Deep homology it's a kind of homology that deep homology refers to, for example, this figure show this very good in a very good way, I think, because, for example, some of the gene networks like Hox cluster, that they are the specific genes that generate the identity of all the segment in animals, in vertebrates, for example, in the anterior posterior, in this way, from your head until your back, the anterior posterior axis. And we look that this is a deep homology because all the animals have this kind of Hox cluster that they have been changed by duplication or other things. But the first homeobox gene that refers to this kind of genes emerged before all the animals. Actually, in this illustration, they show that emerged before in a very deep lineage, like before the division of plants and animals. So it's very old. And as this figure show, this kind of Hox cluster, they are very deep, and actually they can generate the identity of the different segments between in a fly, but also in a human. And we have more than 800 million of years evolution. But also there are another concept, the concept of homology, that refers the typical example of this is we refer to homology that some structures that develop that we look during evolution, but also we look them during the development of the organism, for example, and that we find between the structures a history in common, an evolutionary history in common. So, for example, here the typical example is the limb, our limbs, for example, the forelimbs or the hind limbs. And if we look our evolutions since we were fishes, we look at the different structures that fishes had in their limbs. We develop some structures that they are very similar. Not very similar in the function, because in this case the function was totally different. And in this case, for example, they show a human swimming, but with an arm that is not specifically adapted to swim. But we have the typical, the same structure, and the development underlying this is the same, or very similar. Okay, and oh, this is a little bit messy, but in related to the limb, I am going to focus more in the limb development. In general, in all vertebrates, the development of the limb is very similar. We have, I am not going to enter in details, but in general, at the beginning, very early during the development, there are some inductions that generates two very important tissues that we call organizers. Okay. One tissue is the TPA, the some posterior polarizing, and an apical ectodermal ridge. And one interesting thing about this is that, for example, this CPA emerged in a specific stage and started to induce, by secretion of one molecule, sonic hedgehog, the production of other molecules and the maintaining of the other organizer. And in this way, there are two big tissues, that there are two big organizers that they started to secrete some molecules, and between them, through the generation of new molecules, they start to generate positive and negative feedback. Okay, so I wanted to say that because the positive and negative feedback is very important for a dynamic adaptive system. So, and what is an organizer? An organizer is a group of cells that harbor the ability to instruct other cells into specific states, steering their development into specific organs and tissue. So by the emergence of organizers, during development are very important to induce the generation of other tissues to generate more complexity in the structure. And and the organizer in general generates some molecules that morphogens, for example, that they are very well described for this kind of models that sometimes they say that it's the French flag, because depending on the position and the amount of morphogen, the cells that are going to be here or here or here, they are going to get different identity. And the organizers are very important because, and not, the organizer can be seen as an homology because they are very conserved in a lot of vertebrates, in general invertebrates. I cannot say that in all vertebrates because some of them, they are not, there are not so many experiments with them. But for example, here we can look in mouse or pig or in chicken that the organizers induce the generation of different molecules, and we can look, it's not important which molecule, but the molecules, they have the same pattern of expression, despite they are very different in an evolutionary way. This mouse and pig, they are mammals, but in the case of sheep, that is pretty similar. The divergence of sheep was very, very long time ago. And the same happened with the fingers, the development of the fingers. And so the develop of this depends on the positive and negative feedback of all the molecules that are involved in these processes. And for example, organizers are very important, and one of the typical experiments that are done, we do with to know what is an organizer is to, for example, take one piece of this and transplant that region in another region. And for example, that is one in this experiment that I don't remember the investigator that did before this, but that was done here, and we can look that there are two this is a avian wing, and we look that there are two avian wings, two axes that develop for this insertion. So organizers are very important, and also for example, in the case of, so we know that there are conserved patterns in the limb development by these organizers. And also, for example, there are some experiments that Stuart Newman, their works are very interesting too. He did some simulations trying to replicate the action of this specific zone, a ear, and he could describe this simulation. For example, here he started with an atypical ear, and he stopped the function of the ear at some point during development, and we looked at all these possible to get this kind of a structure that is the humerus, for example. And later in development, you can develop this and so on. And he discovered that with only the action of this organizer, it's possible to reproduce all the structures of fossil, for example, that we look in this case, for example, an ichthyosaurs or some very ancestral structures. But this is very interesting because in general, in vertebrate evolution, we look that only these ancestral vertebrates, these ancestral vertebrates, they have very complex forms. But during the evolution, specifically on tetrapod evolution, not in the case of this wing, but for example, in the legs, we looked at always a conserved patterns of the legs in all vertebrates is to have 5 digits. 5 digits, and this kind of polydactyly or this kind of complexity cannot continue. And I think that is very related with something that you speak about, the constraint, developmental constraints. And sometimes there are some conditions, anomalies, yes, condition or anomalies. I don't like so much to speak about this as a pathologist, because for me it's not a pathology. It's like an state of development. But anomalies that some animals and humans develop polydactyly, but this is not so common. So in general, they are produced, for example, for the use of drugs or the use of the exposition of or some toxics, or for some genetical mutations, but this is not something very common. The common thing is to have these typical patterns of 5 digits. And in the case of organizers also, we can continue having more examples, and what other kind of examples about the organizers is the typical Spearman mangle organizer that, for example, in an amphibian gastrula, one region is the dorsal blastopore lip, and if we take this part and we translate that in another part of the embryo, we can develop 2 embryos, because this organizer is very important to define all the anterior-posterior structure. They define a very important structure that is the notochord that develops the entire body principally. And there are another organizer that is the Hensen node, that, for example, in this case, it's possible to do that experiment between a duck embryo and a chick embryo, very different, but it's possible to induce if it's a region that is in the very anterior in the embryo. And if occur this transplantation good, you are going to see that it's possible to induce a neural tube, the generation of the notochord and a neural tube in the same way to the other. Okay. So very briefly, I wanted to take some concepts that in the previous talk we listened, and based in some concept of a work of Francis. I wanted to say as some conclusions that organizers, for me, I think that they are examples of attractors. Because at some way the organizers change all the direction and all the direction of the molecules that they are around them, and have this capacity of generate a basin of attraction using morphogens. And also, for example, there are other kind of organizers that they are a little bit different during this pharyngula stage, very early during the development of vertebrates too. There are another organizer, the PSM, prosomitic mesoderm, that begin very anterior, but started to move and generate a change in the surrounding space. But it's moving. This PSM is moving from an anterior position to a posterior position, and while it's moving, it's starting to change the mesoderm around and generate this kind of somites that they are very important to the development. And for example, the somites generate after the mass or the different structure in an anterior posterior way. And they generate the vertebrae, the vertebrae, for example. And we can look here that if in snakes, for example, the generation of somites is faster, 4 times faster than the generation of somites in a short body vertebrate like a chick or like a rabbit, etc. So I think that this phenomena is more related to what Francis defined like a limit cycle attractor, because it's something that is changing and is a periodic organizer. So as a conclusion, I try to relate a little bit some concepts of dynamic system applied to embryology. And for example, as a conclusion, we can look that during development of structures, there is a directionality that arises through the natural history. So this conservation or the conservation of the structure and the development of the structure depends on the natural history because they are in general homologies. We look at autonomy of the embryological structure also in their development. We look that also there is a equifinality of forms in some structures and the timing, for example, the philotypic stage during development and evolution. Homologies and organizers could define the conservation of the structures. For example, I think that organizers and homologies, not only the structure that is homology, but also all the pattern, all the molecules that involve, all the reaction involved in the structure during the development can promote the resilience and the robustness of the system. And also with the organizers, and organizers can act as attractors of the system and at the same time define a basin of attraction. And I want to finish just saying this because I put this slide, but I didn't know if I could speak so much about that because it's an open question. What is the anomoplasy? Because anomoplasy that involve convergence, parallelism, and reversion. For example, in the case of birds, of pterosaurs, or bats, they all develop wings, but they are not totally the same because they are based, for example, in this case of an elongation of the digit 4, in this case of an elongation of all the digits, and in this case of an elongation of the entire wing. And or, for example, how long is the neck. And why I put this to put like an open slide is because I think that it's necessary also to take to know what happened in different cases, what happened in different lineages, that they are not totally related each other. And if it's possible, for example, to find something in common between them. For example, we can speak about homology in their organizer and etc. And well, I put this just for a discussion. And thank you so much for your attention. Okay, I'm sorry.
Yeah. No, thanks, Daniela. No, right on time. And thank you for your talk, which has a good audience. And it's time for questions. We should be finishing like right now, but just have like 1 or 2 minutes for questions. If anybody wants to make a comment, if not, Daniela's email is here. Yeah. So you can write to her or write to us as well. So yeah, for leaving time for the other workshops, we will stop here and thank everybody for your attendance, your participation, and yeah, thanks to the organizers, Barbara and ALife people. So wish you a good rest of the conference. So thank you, everyone. Bye.
Bye. Thank you.
Bye.